Thanks for joining me for the fourth and final part of Chapter 21, the immune system, from our Human Anatomy and Physiology text. Today we're going to be talking about transplants, organ transplants, grafts, and then we'll be talking about immunodeficiencies and finally hypersensitivities. So, organ transplants. There's different types of transplants and they're named for the source of the donor material. Autografts are a type of transplant in which the tissue comes from the host's body, from the recipient's own body. So sometimes someone might need a skin graft. We'll take skin from one part of the body and use it to re-epithelialize some other part of the epidermis or the skin surface. But also sometimes much more complex grafts are needed to cover parts of the body after trauma or radical surgeries to remove cancers. We might need some skin and some hypodermis and even some muscle tissue. So all kinds of autografts may be possible. Isografts are grafts between identical twins whose MHC protein complexes are identical, and so it's a very easy kind of graft to accept by the immune system. Allografts are the most common kind of grafts, though, because that's the kind you think of when you hear about somebody having to get a matched donor. We're going to try to find somebody whose MHCs and other markers of tissues are as close or as similar as possible so that the graft won't be rejected by the immune system. And finally, xenografts are very, very important. That's taking animal tissue and grafting into a human. And heart valves are typically xenografts. So the success of the grafts depends on how well matched the tissues are. Autografts and isografts are perfect matches and won't produce any rejection problems. But the most common grafts are allografts, and in that case, we need to match the tissues, but we're also still going to have a tendency of the immune system to attack and reject the tissue, as we'll see. There's a lot of effort going into research to try to figure out how to create xenografts, animals whose organs can be transplanted into humans with less rejection or no rejection. So that's been an ongoing area of research to create animals whose cells express markers that are compatible with human immune systems and so forth, or none. I started reading on that. It's pretty complicated. It's not as easy as we wish. Engineered organs. Some laboratories, bioengineering laboratories, are working on developing functional subunits of various organs like kidneys and so forth in the laboratory, starting from cells from the person's own body. So what can we do to try to prevent that rejection problem of allografts? Well, after surgery and the recipient receives that foreign tissue into the body, something's going to have to be done to suppress inflammation and immune system function in order to prevent rejection. So as soon as we start adding corticosteroid drugs as a lifelong prescription, then we're going to have some side effects. We're going to have a couple of problems, as we'll see. So if you take a corticosteroid drug for a long, long time, you're going to have Cushing's syndrome. The side effects of that kind of a treatment is if you had an overexpression of the hormone cortisol. And so we're going to have hypertension, potentially hyperlipidemia, suppressed immune function, of course, and that leads to susceptibility to disease. All kinds of problems with Cushing's syndrome. So those moon face, unusual fat redistribution in the body. So people that are treated for chronic inflammation or for organ transplants have these problems. We're going to have, again, immunosusceptibility, so antibiotics are going to have to be used to prevent infections in people that are now somewhat immunosuppressed all the time. They have to be much more careful who they circulate with, where they go. I mean, we're all getting used to having to isolate because of COVID-19. People that have had organ transplants have to be careful all the time. Okay. There's been some interesting strategies to try to ameliorate these problems, circumvent these problems by way of developing research technologies. What if we could produce a chimeric immune system in someone's body so that the grafted tissue would be viewed as self by the immune system? So we're going to add some of the the donor's immune system to the recipient in such a way that there's a combined immune system that might allow that tissue to persist in the body. We would have tolerance to that tissue. Regulatory T cells, we said, are a subset of helper T cells that reduce immune function, and they are designed to prevent over-the-top activation of immune responses. Maybe we could produce synthetically programmed T cells that would suppress the immune system in just the case of the transplanted tissue. Xenografts, as we were saying, what if we could produce animals whose tissues would not look foreign to the human body, increasing the breadth of xenograft possibilities instead of just heart valves are a great xenografted tissue because it's largely just collagen, and collagen isn't immunogenic, it's not antigenic, and so it can just be inserted into the body without any need for anti-rejection drugs. Or again, developing organs engineered from someone's own body. Okay, let's move on to immunodeficiencies. Immunodeficiencies fall into two categories: congenital, meaning present at birth, versus acquired. If you already have a normal immune system and then something happens, something goes wrong in your life, and you start becoming susceptible to diseases, new susceptibility, that's an acquired immunodeficiency. So congenital immunodeficiencies, there's a wide range. Virtually anything that you can remember that we've learned about with respect to the mechanisms of innate immunity, as well as especially adaptive immunity. If there's any receptor missing, any ability to express a cytokine missing, you will have some kind of immunodeficiency, and there's quite a varied type of susceptibility that arises in these cases. But the most severe case is one in which there is no adaptive immune response, severe combined immunodeficiency syndrome, the bubble boy disease. You may have seen on television some historical documentaries about this, or you might have even seen a silly show, a Seinfeld episode that was dedicated to this idea of the bubble boy disease. Well, there was actually a boy born in 1971, I believe, who had severe combined immunodeficiency, and it became a very famous, celebrated case, and he lived for a number of years inside of a sterile plastic bubble, large enough, like a room-sized bubble, so that he could have toys and his bed and all kind of things, and people could reach in with long arm-length gloves that were attached to the wall and make contact with him, but he couldn't be out of that bubble, and eventually they had to take him out for the need to do some health care related treatments, and he acquired an infection and died. So yeah, in this case there's no helper T cells, and so there's no adaptive immune response, no humoral immunity, and no cytotoxic cellular immunity. So very, very susceptible to virtually any microorganism that can get past the epidermis or mucous membrane. Other acquired mechanism of acquired immunodeficiency, cancers that crowd out white blood cell synthesis can produce immunodeficiencies. Leukemias are cancers of white blood cells in the bone marrow, and the type of cell that's overproduced, those cells are not functional, and they tend to crowd out the production of other cells in the red bone marrow, and so you're going to have susceptibility to disease, immunodeficiency. Lymphomas are similar, except that the lymphocytes, the B cells, are living in the lymphatic system, and that's where the cancer arises. And there's a couple of classes of actually lymphomas. Hodgkin's lymphoma is a lymphoma, a cancer that produces a solid, hard mass of cancer cells in some part of the lymphatic system, and it can be usually removed surgically without any problem. Non-Hodgkin's lymphoma means every other kind of lymphoma, and there are many different types of non-Hodgkin's lymphoma, and they range in severity quite dramatically. Anyway, the same kind of thing happens. We have non-functional cancer cells, and we have crowding out of the production of normal white blood cells. In the case of lymphoma, eventually the cancer cells will populate the bone marrow, will travel to the bone marrow and metastasize and cause the same kind of problems as leukemias in that regard. All right. Acquired immunodeficiency syndrome, the most famous, oh, I almost used a terrible term, the most famous immunodeficiency, the human immunodeficiency virus causes this situation after years of infection. it attacks helper T cells. So we're going to cripple, again, adaptive immune system. We're going to have a severe combined immunodeficiency like syndrome by action of a virus. It's going to be characterized by some initial findings: night sweats, unexplained weight loss, swollen lymph nodes, and opportunistic infections. Opportunistic infections are ones that that infect the body and cause disease that normally would not be present. Things that normally would be easily conquered by your immune system and would not cause disease in healthy humans. For example, someone with this immunodeficiency might go out in the garden, try to dig in the soil, and some just very common routine fungi that grow in the soil that we are used to having all over us all the time can actually enter the body and cause life-threatening disease because the immune system just can't control anything. Pneumocystis pneumonia. Pneumocystis jirovecii is a protozoa that can cause pneumonia in people with HIV/AIDS. It's actually diagnostic for the disease because it doesn't occur in the normal population. Also, the way HIV was probably one of the things that allowed HIV to first be identified was the prevalence of a cancer called Kaposi sarcoma, skin cancer, that results from a human herpes virus type 8 infection, a very, very rare cancer. And when a sort of burst of incidence of this disease occurred in a region, I think it was in Chicago in 1980s. That began the hunt for this virus that caused immunodeficiency. So the HIV, human immunodeficiency virus, is transmitted in body fluids. That's why in the lab, for example, we treated all body fluids as if they were potentially HIV positive. Get in the habit of using the proper personal protective equipment, because blood, semen, vaginal secretions can transfer the live virus from one person to another. The most common way that the virus is transferred is probably through sexual contact. Blood transfusions that haven't been treated, that rarely happens anymore. There was a time when nobody knew about HIV, we didn't know enough about HIV, and people would go into the hospital for a medical treatment and receive a transfusion that was positive for HIV and become infected. Drug addicts that share needles, they don't have access to a huge supply of fresh needles, excuse me, and so they share needles and they transmit disease. So, how does HIV deplete helper T cells? How does it get into helper T cells? Well, HIV has a coat protein. That means that on the surface of the virus is a cell membrane, essentially wrapped around the coat. The virus genome is covered by a protein coat, and then that is surrounded by a membranous capsule. And one of those proteins on the surface of the virus can bind to CD4 and helper T cells, and that will induce endocytosis, and the virus will be taken up into the cell. Well, this is an RNA virus. That means that the virus particle, that encapsulated piece of nucleic acid, of genes, is in the form of RNA. And so when the virus gets inside the cell, it uncoats that viral RNA genome, and then it has its own enzyme to copy that into DNA. It's called a reverse transcriptase, and that's because virtually all organisms store their genome as DNA. All eukaryotic organisms, I should say, store their genome as DNA, and then when they want to produce some protein, they copy or transcribe some of that DNA into the form of messenger RNA, and that RNA is used to translate proteins. And there is no way to go the other direction, but this virus uniquely has what's called reverse transcriptase, so it can take a piece of RNA as a template and produce a DNA copy of it. Okay, so now we have a genome in the form of DNA from the virus, and there's enzymes in the virus genome also to insert that into a chromosome of the host, just random insertion into a chromosome, and that genome of the virus in the eukaryotic cell is now called a provirus, in this case a helper T cell. And so then that provirus will then direct the host cell to make viral RNA and proteins. So messenger RNAs will be made just like a expression of any gene in the nucleus of the cell, and the virus, all those proteins will assemble along with new viral RNAs and produce new virus particles that will go on and infect other cells in the body and kill the host cell. The one interesting problem is that the provirus, though, can remain silent within helper T cells for years, for many years, decades even. And so instead of killing a cell, it just lies quietly within a chromosome in the cell, and that is a significant problem because we have very effective antiretroviral drug therapies now that can rid the body of any active HIV virus. But we can't target or discover the location of all provirus in the body, so that even though someone has been rid of the multiplying viral replication competent virus, there may still be an infection that arises, an outbreak later in life. So what happens when we start killing off these T cells, these helper cells? As the HIV virus kills more and more T cells, then the immune system starts making more helper T cells. The bone marrow, the red bone marrow, and the thymus rev up and produce helper T cells at just an astonishing rate, day after day, year after year, and eventually, after a decade or so of this war between the virus killing the helper T cells and the production of new helper T cells, the immune system fails, and the T cell population drops precipitously, and the person becomes completely susceptible to opportunistic infections. Anything that can enter your body can basically colonize your body. Even the virus actually enters the central nervous system and causes dementia, causes damage to the brain. So how can we combat this? Well, we have successfully examined all of the steps that the virus uses to, when it enters the cell, to uncode itself, to reverse transcribe its RNA into DNA, insert the DNA into the chromosome, produce all of its gene products, and so forth. So scientists have found drugs that can block any one of those different steps, and if you combine drugs that can block multiple steps in the virus life cycle in cells, you can successfully stop its replication, and eventually it will die out. So what are some examples? Fusion inhibitors, so that the HIV can't enter into the host cell and never can find its way in, and if it can't find a cell to replicate, eventually it will degrade and die. Integrase inhibitors that block the viral genome from inserting itself into the host chromosome. Reverse transcriptase inhibitors prevent the production of DNA form of the viral genome that's needed to insert into the host chromosome. Protease inhibitors prevent the maturation of virus proteins. Some of the proteins are actually translated as double and triple length gene products, these sort of concatenated proteins that need to be trimmed by special proteases to produce the mature proteins. Actually, there's actually a lab in University of Rochester that has been researching actively multiple ways to try to prevent the transmission of HIV, and one of the things that they have discovered is a way of producing a vaginal gel that will prevent the HIV virus from getting a foothold in the body. And they're still working on perfecting that. And this has been a while ago since I was familiar with their work, but I assume they've made even further progress with that. So that's our story about immunodeficiencies. We now move to the opposite end of the spectrum. What about cases where the immune system is overactive, doing some things that we really wish it wouldn't do? Autoimmune diseases. So in some cases, the form in which the immune system does some excess things that we don't want it to do is in the arena of attacking our own body. Losing the ability to discern self versus non-self. For example, the immune system may produce antibodies against antigens of our own body, autoantibodies they're called, and sensitized T cells, cytotoxic T cells, may be going around and destroying cells of tissues of our body. So there's a couple of ways that we could have an autoimmune disease. Here's a list of autoimmune diseases, and I hope you don't find it off-putting, but instead fascinating. Almost all of these we have talked about in various chapters. Type 1 diabetes mellitus is an autoimmune attack on the islet cells of the pancreas so that there's no longer any insulin production. Rheumatoid arthritis we talked about when we talked about joints, and a chronic inflammatory disease in which the joints are destroyed, the joint articular cartilage is destroyed in there, and breakdown of the joints. And psoriasis, we didn't really talk too much about psoriasis. We could have probably emphasized more in chapter 5 on the skin. It's an autoimmune inflammatory condition of the skin, but it also has multiple sequelae, including arthritis, a bad arthritis condition. Multiple sclerosis is that autoimmune disease in which the myelin sheath on myelinated neurons is attacked, and eventually that causes all kinds of problems with nerve communication in the nervous system and so forth. Lupus is an autoimmune disease in which there's autoantibodies against a whole range of different self-antigens. Inflammatory bowel disease, Crohn's disease, autoimmune diseases in which there's chronic and severe inflammation of the bowel. Excuse me. Addison's disease we also talked about. It's that disease in which the adrenal cortex is targeted. We don't have enough cortisol, or I can't say the word, I have a little hiccup. It'll come back to me in a second. Graves' disease is a disease of activation of the thyroid gland, amazingly. Some autoantibodies that bind to the thyroid stimulating hormone receptors on the thyroid gland crank up the production of too much thyroid hormone. Hashimoto's disease, in which autoantibodies are attacking and causing destruction of the thyroid gland and loss of thyroid hormone production, so hypothyroid. Myasthenia gravis, we have attack on the acetylcholine receptors in the neuromuscular junction of skeletal muscles, and it causes progressive muscle weakness because we can't activate muscle contraction. Glomerulonephritis causes damage to the glomeruli in the kidneys. And pemphigus vulgaris I put in there just because I came from a background of research in the skin, and it's a terrible blistering disease of the skin in which some of the membrane proteins that anchor cells of the epidermis are missing or are blocked by antibodies, and there's just splitting of the layers of the skin and fluid filling these huge vesicles, and it's a terrible disease indeed. So that's just a list of some very interesting autoimmune diseases, and a lot of these weren't even known to be autoimmune diseases. They were treated for symptoms for many, many years without knowing their etiology, and now we're starting to realize that they are in fact autoimmune diseases. So what can we do about it? Well, treating with anti-inflammatory drugs and immunosuppressant drugs. So a lot like what we have to do for people with organ transplants. And so, as you know, then we're going to have a couple of problems. We're going to have susceptibility to infection. We're going to have the potential for side effects from chronic treatment with corticosteroids, namely Cushing's syndrome. Aldosterone, okay, there we go. I knew that word would pop up. For Addison's disease, aldosterone insufficiency, as well as cortisol. All right, how can we do a better job, a more targeted action against autoimmune diseases? Well, that's been some pretty successful stories recently, more recently, of antibodies that are designed to bind to and inactivate very important inflammatory mediators in some of these diseases, like tumor necrosis factor alpha. And in Crohn's disease, antibodies against tumor necrosis factor alpha, or against the receptor to tumor necrosis factor alpha, and more recently to anti-interleukin 12, have been very effective for many people in reducing that inflammation in the bowel, and also in psoriasis. So, and it may be many more as well. Also, antibodies against co-stimulatory molecules have been successfully applied, in which we can prevent some of the activation of B cells and cytotoxic T cells by helper T cells, or activation of helper T cells by antigen-presenting cells. course, again, this is a very fine line, a balancing act between suppressing the immune system enough to ameliorate these diseases without becoming susceptible to everything that comes down the pike. There's a lot of research, of course, going on. We're learning more and more about the immune system, and we are looking for more ways, the scientific community is looking for more ways to combat autoimmune diseases without the side effects and risks. So maybe making sort of engineered regulatory T cells that will help suppress particular activations that cause disease, or self-tissue targeting antibodies or cytotoxic T cells, vaccines to promote self-tolerance, providing antibodies against self-reactive immune cells so that then the immune system will attack those cells and destroy them, and they won't have this ongoing inflammatory or self-attacking disease. It's interesting too, some of the theories on how autoimmune diseases arise, the etiology of autoimmune diseases. There's been a lot of theories that have been put forth, and some of them have been pretty well convincingly established. Some of them are purely epidemiological analyses. For example, there's a genetic component to autoimmune diseases. It runs in families. Certain MHC classes, some people with particular MHC types, there's sort of like subgroups of different MHC types that people may have. Everyone's MHCs are unique, but there are groupings, and certain MHCs tend to be associated with a higher incidence of autoimmune diseases. Smoking, there's been a direct correlation been made between smoking and a much higher incidence or risk of autoimmune disease. Urban dwelling. So it's assumed that pollutants may act in a way similar to cigarette smoke in promoting autoimmunity in people. The basic bottom line for why we have autoimmunity is, besides just being genetically predisposed by MHC type, is the defective elimination of self-reactive lymphocytes. We talked about T cell education or maturation in the thymus and B cells in the red bone marrow. And what we need to do is eliminate all of the T cells and B cells whose receptors would recognize a self-antigen and only retain those cells which would recognize foreign antigens coming into our body. Well, it's a subtle thing. There are some chemical sort of epitopes or recognition sites that may be very similar between self versus non-self, and if we have retain some lymphocytes that are a little too close to recognizing self as foreign, they may be revved up in some way by some stimulus that eventually at some point in time leads to an autoimmune disease. Another way, foreign antigens might come into the body in the form of an infection that are similar, again, somewhat similar to epitopes on a human protein, and after the successful eradication of the infection, we might wind up with some components of the immune system that now recognize some self protein as foreign, because of that overlap, that cross-reactivity it's called. Sometimes there could be generation of new foreign-looking antigens if we have a gene mutation, so the protein is still being expressed in cells, but it's changed from the way the normal protein used to look. Now we have new epitopes potentially that the immune system has never seen. It wasn't present during the maturation or selection of lymphocytes in the thymus and bone marrow, and so now we may have some of our formerly foreign antigen recognizing cells are going to recognize this newly mutated protein. Also, it's possible that a denatured protein after an injury to tissues or possibly exposure to a hapten might induce an immune response that thereafter might cause attacking of the original native protein because of the similarity between the two. So that's another theory on etiology of autoimmune diseases. Traumatic injury, there's a correlation, a correspondence between traumatic injury and the later development of autoimmune diseases. That's been established epidemiologically. Why is that? Some theories include a profound activation of inflammation to a level the body has never seen before, and it might cause just such an activation of immune system that we wind up with some accidental production of some immunity against self. Exposure of tissues that are normally kind of sequestered away from the immune system, now exposed to myriad lymphocytes that may actually have receptors that would tend to cross-react to some of those antigens. So anyway. Oh yeah, an interesting one. What if some cells from the mother's body somehow cross the placenta and take up residence and form a colony somewhere in the fetus and survive to birth, and maybe as the immune system develops, it may sometime later in life discover those cells and recognize them as foreign and attack, and then that might actually sort of cause cross-reaction with the body itself. There may be much more subtlety to that than I have read about, but it might be somewhere. If you're interested in that, take a look and see what is going on. Looking for a research article on etiology of autoimmune diseases and the transfer of cells from mother to fetus or from fetus to mother. So that's our story on hypersensitivity. Let's just quickly look at 4 types of hypersensitivities. They're classically grouped into four sort of camps. There's actually more subtle distinction between some of the examples within these different groups 1 through 4 that we're going to talk about, but we're just going to look at those 4 groups together right now. There are 3, types 1, 2, and 3, that are caused by antibodies, and then there's type 4 that's produced by direct activation of T cells. All right, so type 1 hypersensitivities are called immediate hypersensitivities or acute hypersensitivities, and the technical term for it is anaphylaxis. Anaphylaxis is the correct term for this type of reaction. When people say anaphylaxis, though, oftentimes what they're talking about is a systemic anaphylaxis in which someone is in risk of dying from it. So what's going on there? Well, we have a primary immune response called sensitization that's going to wind up producing a Th2 response. Helper T cells are going to mature and become activated that secrete interleukin 4. And so when the B cells ultimately are activated by those helper T cells by interleukin 4, or by the cytokines produced by those Th2 cells, we're going to wind up with immunoglobulin type E production instead of immunoglobulin type G. Well, the immunoglobulin E's will attach themselves after the disease is eradicated, whatever the immune system was responding to, and attach themselves to mast cells and basophils, and secondary exposure will result in inflammation. So this could happen at the local level. You could have an allergy where you have local swelling. It could be in your respiratory mucosa. You could have hay fever where you have pollen coming in and activating this response, and you have the runny nose and the itchy, watery eyes. You could have asthma causing bronchoconstriction and inflammation in the lungs. You could have it in your GI tract, eat some food that you're allergic to, and it's going to cause inflammation in your GI tract. Some unfortunate experiences there. And it'll be treated, we usually treat it with antihistamines for most people, but in worst cases, we might need some corticosteroid treatment during a bout of a local hypersensitivity. It could be systemic. If you eat an allergen or an antigen that produces this kind of response, and it'll go from your digestive tract into the blood, and now it can circulate. That can cause so much vasodilation that total peripheral resistance goes down, and you wind up with circulatory shock, potentially not enough blood pressure to drive circulation. The heart has nothing to work with, and you could die. Treatment, the EpiPen. We've talked quite a few times about the EpiPen. So epinephrine, in a form of an injectable device, can be carried by someone who knows they have such an allergic sensitivity, a bee sting sensitivity, who knows, or a nut allergy or something like that. And we can bronchodilate them and at least help them survive until the immune system settles back down. Here's a little graphic showing the process. The primary immune response, in which the antigen enters the body. We have clonal selection and proliferation and maturation of B cells. Antibodies produced. The antibodies happen to be immunoglobulin type E. And after the immune system response, then those immunoglobulins, by way of their stem region, attach to mast cells. secondary immune responses thereafter result in antigen binding to those antibodies on mast cells, and the mast cells degranulate and release histamine and cause all kinds of inflammatory effects of histamine, vasodilation and leakiness of blood vessels, and mucus production in the airways and so forth. Bronchoconstriction, smooth muscle in the airways contracting and making it very difficult to ventilate the lungs. Type 2 hypersensitivities are called cytotoxic reactions, and they're characterized by antibodies that will bind to cells, and when they bind to the cells, they'll activate the complement system, or phagocytosis. Will have destruction of cells by phagocytes or complement mediated lysis because of some antibodies. For example, if you receive a blood transfusion from someone that has antigens on the blood cells for which you have an antibody in your circulation, your antibodies will bind to the surface of those red blood cells and activate complement, and you'll have hemolysis. Sometimes certain drugs have been shown to cause an immune response against platelets. So if the drug that's being used completely unrelated to platelets, a drug that's in the circulation because we've given an oral dose or something like that, and that happens to bind to the platelets and creating an immune response that then thereafter makes the platelets the target of the immune response, then we could remove or lose all those circulating platelets and have thrombocytopenia. Graves' disease actually is considered a type 2 hypersensitivity, although again, if you expand out into I think there's 7 typically defined types of hypersensitivities. Graves' disease would be a type 5 hypersensitivity, but it's often classically put into this type 2 category where we have antibodies binding to cells, but in this case binding to a receptor and activating the thyroid stimulating hormone receptor on the thyroid gland. Type 3 hypersensitivities. These are ones in which there are antibodies widely distributed in the body, then they're present chronically. So you have this constant exposure to an allergen, constant production of antibodies that are circulating, and antibody-antigen complexes in the body all the time, and there's excess antigen so that we never form this huge lattice and precipitate of antibody-antigen complexes. They're always present, and they tend to lodge themselves in blood capillaries and activate neutrophils, and neutrophils release all kinds of inflammatory mediators, and we have inflammation happening, tissue damage, and especially in the microvessels. It could happen in the kidneys. It would cause kidney damage, failure of the filtration mechanism apparatus. Could happen in the lungs. Farmer's lung is a classic example of type 3 hypersensitivity, in which chronic seasonal exposure to something having to do with hay, for example, will cause eventually permanent destruction of the lungs through all this chronic inflammation. Lupus is a disease in which there's antibodies, autoantibodies being produced against all kinds of antigens. There's circulating antibodies all the time, and there's inflammatory destruction of organs. That's the problem. Type 4 hypersensitivities are called delayed type hypersensitivities. Now, type 1, 2, and 3 that we just talked about are all antibody-mediated hypersensitivities. This one is direct activation of helper T cells, typically by a hapten that comes into the body, binds to a protein in the skin, and that directly activates T cells. And the T cells start releasing cytokines that activate macrophages and activate cytotoxic T cells, and they're going to start attacking and destroying cells of the tissues and have inflammation in the skin and skin damage oftentimes. So allergic contact dermatitis, you may be allergic to poison ivy, for example, or other allergens that cause this kind of days later exposure, and then days later this eruption of vesicular inflammatory nodules in the skin. Very, very itchy as well. The tuberculosis skin test, if you go to apply for a job and an employer is thinking of hiring you, one of the things they'll be required to do is give you a tuberculosis test. That was instituted by our Food and Drug Administration many years ago to control the spread of an incidence of tuberculosis in our population. It's been very, very effective. So everyone that goes to work gets a TB test. So again, these are haptens that are causing response. There's a couple of things to note about the immune system that are interesting. The emotional state, the fatigue status of the body, those things amazingly can impair the immune system. So if you're run down, yes, you're more susceptible to disease. When do you get colds? Right around December, when you're trying to do 5 papers and study for 4 final exams and not getting enough sleep and you're stressed out, yeah, you're susceptible to disease. Dietary considerations, if you're not getting good nutrition, then you're more susceptible to disease. Specifically, vitamin D has been associated with susceptibility to disease, interestingly. With aging, the immune system changes. Someone has mentioned acquiring allergies with progression through life, and that is definitely a thing. But also, as you age, your immune system begins to become more deficient and less efficient, but also more trouble-prone, more likely to develop autoimmune diseases later in life. More cancers arise. Many tumor cells are produced throughout your life, and they're destroyed by natural killer cells and cytotoxic T cells. And in the absence of an efficient anti-tumor response of that type, cancers are much more frequent because they just win the battle. A tumor is basically a winning a war against your immune system to conquer abnormal cells before they grow into a large enough mass to really be able to hold their own. So that's our story about the immune system, and I hope you have learned a lot about the immune system and have a lot of good takeaways from that, and we'll come back together again and talk about the respiratory system next.